السلام عليكم. بنرحب بحضراتكم بحضور مجموعة من السيمينارز free international seminars organized by Nanotech Solutions Canada. بيشرفنا في المحاضرة الأولى الأستاذ الدكتور وليد توفيق، رئيس قسم تطبيقات الليزر في القياسات والكيمياء الضوئية والزراعة بالمعهد القومي للليزر جامعة القاهرة. We had some attendees who have registered from non-Egyptians. So can I know if all the attendees understand Arabic, or we have just English speakers here? Do we have any English speakers only, or some attendees who cannot understand Arabic?
Hello, assalamu alaikum.
Alaikum assalam.
I am Saifullah Jamali from Pakistan, University of Sindh Jamshoro. So I cannot understand Arabic. I can only understand English.
We are welcoming you, so we will try that to make all the presentation in English. We will start now in a minute. We have Professor Walid Tawfik, Professor of Laser, Spectroscopy and Ultrafast Lasers, National Institute of Laser Enhanced Science. Professor Walid Tawfik is the chairman of Department of Laser Application in Metrology, Photochemistry and Agriculture, leader of Laser Spectroscopy and Ultrafast Lasers Group, and the head of the Central Lab at the National Institute of Laser at Cairo University, Egypt. So let's start. We are welcoming Professor Walid Tawfik to start the seminar.
Okay, Assalamu alaikum wa rahmatullah. Thank you very much, Dr. Ihsan, for this brief introduction about me and about my job. In fact, I had modified the title of the my talk little bit. I add the word nanotechnology little bit. I introduce something nanotechnology to accommodate the aim of this company, which is one of the leader companies in the world about nanotechnology. I hope that everyone can understand my my lecture in a good language. I think English is can be accommodated for everyone here in our group. Please, if any of you have a problem with English, you can raise your hand. I will try sometimes to switch to some few Arabic words for more explanation if possible, but I will try to focus on English because this is one of the most popular language worldwide. Okay, about recent applications of laser spectroscopy and nanotechnology. In fact, nowadays, as you know, there are many applications of laser spectroscopy in many sciences like material science, environmental science, medical applications, and also there are many applications of nanotechnology in this science as well. In these lectures, I will try to show you one or two applications of combination of laser and nanotechnology, for example, in medical applications. Also, we'll see how laser spectroscopy and laser technology can be used to overcome the climate changes And on the other hand, we will see that laser spectroscopy, one of the leading methods which can be used to discover new technology in spectroscopy, to discover new materials, or we can generate new materials as well. This is the outlines of our talks today. I hope that I can cover all of these points. I'm not sure, but it depends on your background. Sometimes I will go faster, and sometimes I will slow down. I hope that you will not be feeling any boring with my talk. I hope so. Maybe Dr. Hassan gave a brief introduction about me, but I would like to say that nothing came by luck. I mean that you have to do a dedicated efforts day by day and year by year to learn and to grow up. In fact, I have dedicated all my efforts and maybe all my entire life to gain experience in my field of laser spectroscopy. I started my trip to this favorite specialist for me maybe since 1994, after graduation, at which I started my master degree at Cairo University in the field of laser spectroscopy. Then I switched to Germany to earn my PhD at Technical University. After this, I tried to do my best to communicate with most of the famous group worldwide to gain experience wherever and whatever these group are existing in United States, in Boston University in South Korea, maybe Max Planck Institute. Of course, I have a good communications with Max Planck. I spent a lot of time over there from time to time. Poland, also maybe some countries did not mention this small brief about me. Something in Japan and Pakistan, maybe also Canada, and some countries in Europe. Nothing by luck, and I will say to you that none of these opportunities was open for me. I contacted the leaders of this group and explore my experience to them that I can grow up their group in their specialist of laser spectroscopy, and from my experience maybe I can enhance their output. In fact, after Germany graduation in 2000, I learned I have to make my way if there is no opportunity. So that don't wait for an open position. Take your position. This is my advice for all the young scientists. Maybe the questions will be opened after my lectures, please. This is brief about my my publications. In fact, since 19 centuries, the beginning of 19th centuries, Cairo University is well known university worldwide. Maybe the first university in Middle East. Maybe I don't remember exactly, but I think so. Then my institute was the idea of initiated with Professor Lutfiya Nadi almost during my graduation, and she gathered us to an institute of laser, and we were a part of College of Science. Then we separated from College of Science as a separated institute. Now I will show you some of the equipment which you can find in our institute, like electron microscope, lasers, different lasers, YAG lasers. This is continuum leg lasers, but very short time, something like 5 nanoseconds. And we have complete BLD laser, pulsed laser depositions, which we can use to generate some nanoparticles. This is a picosecond laser. We have femtosecond. And we have coating unit. Also, we have another equipment which there is no time to explore, because maybe you have different backgrounds. I will start with what's meant by laser and laser main parts very briefly. Laser is light amplification by stimulated enhanced emission of radiation. Briefly, you can imagine that laser is a very focused light in one direction, and it is monochromatic, just one light, one wavelength, with many parts. One of the common most important property is the photons of this laser are synchronized. Synchronized means that all of the photons are generated in the same time, and they have the same energy, the same frequency, the same directionality, or everything together. Of course, each laser type has different wavelengths and different powers, depends on your applications. But one of them also very good property of the laser that bandwidth is very small, and as I said, it is coherent light. How we can generate laser? Very briefly, let me remind you in the beginning of the human beings on the earth. In the beginning of human being, they were thinking of how we can store water, because water maybe from rain or whatever, they dig some wells to store water, just 2 walls, and there is a space between these walls. This is the wheel, and they really store water. After some 1,000 years, they discovered the electricity, and they said that okay, we may store electricity by having two sheets of metals, and these sheets of metals has different polarity. This is a capacitor at which they can store some charged particles. Then after some times they said, okay, we stored the water and we find a way to store the electrons or electric field. What about the light? Can we make the same ideas as we store the water and the electricity? Can we store photons, for example? They said, okay, if we have two mirrors, what about if we bring a photon inside these two mirrors and we close these two mirrors, and this photon is sweeping, go back and forth between these two mirrors. Can we store this photon? Theoretically, yes, they can store photon. But after a while, after a few seconds, the photon is absorbed by water in the air, of course. They try to make these two walls from very clear mirrors in a vacuum to minimize the problem of absorption. After a while, they said, okay, let us try to enhance these photons, because by time, maybe the energy of these photons going to be absorbed. They invented something called active medium. Active medium is just an active sample at which the molecules and atoms of this sample can be excited to very high excitation levels. Then after a while, these very high excitation levels goes down to zero, almost zero ground state, and there is emission of radiation. This is called stimulated emission of radiation or laser action. This is the bumping. Bumping means excitation, and this is excited states, and this is metastable state. You can read about this if you if you interested in this phenomena. And after a while, they enhance the stimulated emission of radiation to have all the emission in the same directionality. Of course, the one of the most important rule in this idea is Maxwell-Boltzmann law. Maxwell-Boltzmann says that if we have ground state atoms at room temperature, most of the atoms are in the ground state. If you excite the atoms, maybe 5% or 2% will be excited, but the most of the population of the atoms will be in the ground state as well. So as well as we increase in temperature of the sample, the number of population maybe will distribute according to the energy, but most of the population will stay also in the ground state. But to have very powerful laser, we have to make something called population inversion. Population inversions بالعربي يعني انقلاب سكاني. انقلاب سكاني يعني نخلي كل الاتومز اللي موجوده في الجراوند ستيت موجودة في ال excited states. So that we can use this formula for Boltzmann distribution function. And for example, at room temperature, if you look at room temperature, you will see that only one for every 1 billion atom is excited, or 4 atoms are excited for every 1 billion atoms for 300 Kelvin. So that only four atoms are excited, but most of the atoms, most of the atoms, 1 billion atoms, are still in the ground state. So that to excite to more number of atoms, of course, you need higher than 300 Kelvin. They use Einstein relations to calculate the probability of how we can excite these atoms. I think the video is not working. This is a video. Something wrong with the video. Okay, no problem. Anyway, and they invented two systems to make lasing, 3-level systems and 4-level systems, at which we can excite the atoms to higher excited states to make population inversion, and from this higher excited state we can generate photons, very powerful photons. I don't want to go through my equations and more details about this, because this is another lecture. I just give an idea, just brief idea about how we can generate lasers. This is, for example, if you use the active medium as yttrium aluminum garnet, you will have YAG laser. If you have argon as exciting medium, you will have argon laser. If you have helium, you have helium-9 laser. If you have CO2, you have CO2 lasers. So most of the type of the laser depending on the active medium. And each active medium, for example, argon, has specific wavelengths at which we can generate lasers. This is a brief idea about how we can generate. Of course, we have diode lasers, at which we have N-type and P-type, and instead of exciting the electrons, we are exciting photons. Important point for beginner researchers that forget about geometrical focusing of the laser. Maybe in theory we draw the laser beam after a lens as a point, focal point. It is not a point, really it is not a point. The focal point is a volume of focus region at which there is a width and length. Why I mention this here? Because if you have a sample, you have to have sample dimensions within this region to have a homogeneity of the laser intensity on the sample. Of course, there is equations at which you can calculate what is the width and what is the depth of this focal region. This is because of the lens aberration. Let us go to the applications now. One of the important applications this night, I will tell you about LIPS. LIPS is laser-induced breakdown spectroscopy, which is a type of atomic emission spectroscopy at which we use a very intense laser to be focused on the surface of the sample to generate plasma. And by analysis emission of this plasma, we can get all the information about this sample. Of course, I did not mention which type of samples we can use. The magic of this method, we can use this method on solid, gas, or liquid. Of course, there is different mechanisms of interactions, different from gas, liquid, solids. Some, for example, there is no reflections from gases. There is no melting from gases or liquid. Melting only happen in solids. So it depends on the mechanism of the interaction, but laser can interact with all type of samples. الثلاث حالات للمادة الغازية والصلبة والسائلة احنا بنقدر نستخدمها مع هذا التكنيك. استأذنكم الإخوة اللي دخلوا حديثا الأسئلة بعد نهاية المحاضرة. شكرا لو سمحتوا. Okay, let us see how the laser interact with the sample. For example, if we have a solid sample, the laser will maybe has a few reflection, but most of the laser will be absorbed and warming up to generate plasma. Plasma is a combination of electrons and ions and very excited atoms. The emission of this plasma can be detected using some spectrometer and ICCD camera. After some time, the temperature of the plasma will cool down, and you will find some fragmentations. LIPS can work with atomic and molecular emission spectroscopy. LIPS can be qualitative and semi-quantitative. Maybe we can say semi-quantitative. LIPS can be real time and can work, as I said, with gas, liquid, and solids. Of course, one of the most important benefits of this technique, it is very cheap. You are not using any consumable materials. There is no loss of your samples. There is no any aggressive chemicals. There is no any danger problems with using lasers if you follow the well-known precautions. If you use LIPS technique, we will focus the laser on the sample, and the emission of the plasma will bring some emission lines, and by analysis these emission lines we will know the composition of the sample. Of course, the temperature and electron temperature and electron density can be known using some equations, using Boltzmann plots. I will not go through plasma measurements because maybe this is out of the scope of this lecture. But just mention that the plasma is generated after the start of the laser, and in the beginning of 100 nanoseconds there is continuum emission. After sometimes there is ionic lines emission. Maybe after 1 microsecond there is atomic lines emissions. After 3 microseconds there is molecular lines emissions. Why this happen? I will say to you, it is a kind of scenario. The scenario is the following. In the beginning, the plasma is very hot. Most of the plasma composites representing the composite at which it is generated. So the emission of the ionic lines is dominant emissions. After some times, the hot ions can attract some electrons, and it will generate excited atoms. The excited atom emissions can be existing after something like 1 microsecond delay time. After some times, these excited atoms relax and recombination can happen. For example, if you have a hydrogen atom and excited hydrogen atom, they can recombine again, and you will find emission of H2 molecule or H2O molecule. This is molecular emission after something like 3 microseconds. This is most of the equations are not visible in these versions. I don't know, something happened during the conversion of this file. Some at the Blackboard conversion. I'm sorry about this. I will show you some of the recent output of our experiments using picosecond laser. We analysis plasma profile of copper. Dr. Rania, please, the questions after the lecture. دكتور رانيا لو سمحتي دكتور رانيا لو سمحتي الأسئلة بعد المحاضرة. دكتور أحسان، أرجو من دكتور أحسان تنوه إن الأسئلة.
Dear attendees, please, we will postpone all the questions till the end of the lecture. If you please, we will make some time for discussion at the end of the lecture. Thank you all. Okay, Dr. Walid, sorry for this. You can go on.
Okay. Now we try to analyze the plasma of copper. This is I will show you. This is science for science application of laser spectroscopy. This is science for science. In this real science application of plasma spectroscopy, we discuss how the plasma is generated with time in picosecond resolution. Other papers, we we used, maybe next week I will show you. We use also femtoseconds. And we detect the plasma temperature change with the laser energy and laser wavelengths, and we get uh, the values of the electron density and electron temperatures using this technique. Of course, as I said, the last paper, this is basic science application. Now let us see how we can use laser spectroscopy in medical application. For example, أنا عشان الناس ما تملش هحاول من وقت لآخر أنوع الابليكيشنز من بيزك ساينس لماتيريال لانفايرمنتال لميديكال عشان ما يحصلش الناس ملل. One of the most problem we face in Egypt is cancer. In our group, we started something like some years ago to use nano technology and laser technology to treatment breast cancer, for example. We use we make induction of cancer in real rats samples, and we used the treatment of photothermal and nano gold to treat this cancer problem. Of course, there is two types of tumors. This is benign. Benign, this means that it is not cancer, and malignant. Malignant means that it is that the tumor is cancer. Cancer is well known to be worldwide problem. It caused in 2015, for example, almost 9 million deaths. This is maybe more than coronavirus problems. Cancer is worldwide problem around the world because of the many causes. In fact, there is a statistical distribution about the how much the men and women infected with this kind of cancer, but for breast cancer, most of them are women. For example, we use nanotechnology at which the very small dimensions of materials in the order of less than 100 nanometers were used, and we used as photosensitizers to absorb. We used gold as a absorber for photosensitizer, absorber for a specific wavelengths of laser. The procedure is as follows. We inject after preparing of gold nanoparticles, we inject the gold inside the tumor of the sample, and we irradiate the sample, the cancer, with laser irradiation. This is a preparation which well known is well-known method for preparation of gold nanoparticles. And this is a procedure at which we inject the nanoparticles in the breast cancer, and we use laser specific wavelengths, 800 around 800 nanometers, to be absorbed by the gold nanoparticles, and it generates heat and free radicals of oxygens, which eliminate the tumor. Of course, heat and free radicals of oxygen are aggressive to all the tissue. But in this treatment, there is two benefits. Laser alone, you cannot select the good cell and the bad cells. And the nanoparticles alone, we found they are not very effective. But both of the technique together, the efficiency is so much. Efficiency is something like after 1 week or some maximum 2 weeks, the breast cancer totally disappear. This is one of the benefits of using laser and nanotechnology. For environmental, to eliminate the problem of climate changes due to pollution, and to overcome this kind of pollution, which generate problem of global warming and increasing the global temperature. We tried to analyze soil and water using laser spectroscopy. For example, soil in Sahara deserts, many samples were analyzed using LIPS technique, and we found, of course, different elements in these samples, and we build a calibration course especially for the toxic elements and heavy elements like Fe, and maybe copper, and other toxic elements, aluminum, for example. And we analyzed how much elements in these soil samples. One of the most benefits you have to put in the mind, انت لازم تبقى فاهم ان الليزر ميزته عن ميزة التحاليل الكيميائية. Laser can finish the analysis in few seconds. Chemical analysis takes something like 1 or 2 days and consuming a lot of samples. Laser has no any consuming of your samples. So that if you have a precious sample from mummies or architectures or biological samples, it will not you will not waste your sample. this is application of LIPS technique in photo gypsum waste treatment. Photo gypsum waste treatment is one of the wastes of phosphor production for agriculture. واحدة من السماد اللي احنا بنستخدمها عندنا في السماد بتاع سماد الأراضي، استصلاح الأراضي، ولقينا الفوسفو جيبسون دي، المادة دي واحدة من المواد الأساسية عندنا في الويستس بتاعتنا بتاعت استصلاح الأراضي. المشكلة الأساسية إن الفوسفو جيبسون دي بيبقى بتحتوي على نسبة من الفوسفور واحنا عاوزين نقللها او نعمل لها مونيتور. One of the main problems that we want to monitor the concentration of phosphor in this material of phosphogypsum, so that we analyze. This is the how much phosphogypsum you have in factory, for example. And you can imagine how much phosphorus can be found in this mountain if it is available, so that we want to save phosphorus to be wastes in this material. So that we use LIPS technique to analyze the content of this sample, and we calculate the plasma temperature and electron density, and we made calibration curves, and we calculated how much the phosphorus content in these materials. Let us return back to medical applications. In these applications, we used LIPS technique to make identification of bacterial samples. البحث ده كان تمهيد ان احنا ممكن نستخدم ليبس لاكتشاف الفرق ما بين بعض الفيروسز والبكتيريا، لانتشار الفيروسز والبكتيريا دلوقتي في واحدة من الحاجات اللي احنا بنواجهها في العصر الحالي زي الكورونا والفيروسز المختلفة. عشان كده استخدمنا ليبس تكنيك في هذا المضمار. We used LIPS technique to distinguish between different types of bacteria. This type of bacteria called Escherichia coli. This coli bacteria dimensions something like the length of this bacteria is about 1 or 2 microns, and the width about half microns. The other type of bacteria called Micrococcus lotus with dimensions almost half microns diameter and length about 2 microns. It is spherical shape, and we want to distinguish between these two types of bacteria. We used laser samples, laser technique to investigate these two types of samples. We found one of the most interesting things, that some of the elements are associated with the membrane of these cells. وجدنا إن بعض الأملاح موجودة على جدران خلايا هذه البكتيريا. حد هيقول لي طب ايه علاقة بالاملاح والبكتيريا؟ البكتيريا عشان تعيش لابد لها من بعض الاملاح، والممبرين بتاع البكتيريا ده بيسمح لبعض الاملاح بالمرور، ما بيسمحش لبعض الاملاح التانية بالمرور. فكونسنتريشن او تركيز الاملاح بيبقى مختلف في كل بكتيريا عن التاني. ده وجدناه عمليا ومثبت علميا، وبناء عليه قدرنا نميز between two types. We have differentiate between two types of bacteria, type A and type B. We found some elements in this bacteria and other elements in the other bacteria. As I said, these elements are primary elements in the membrane of the bacteria to be survived. طبعا we made some statistics about the distribution of these elements and level of concentration, and we distinguish between the two types of bacteria. Maybe in future we can distinguish between types of viruses. Maybe we will try, inshallah. Maybe I will finish with one of the most important applications of laser, which I see it is very benefit these days. Nowadays, one of the problems we have faced that there are many fake medication in the market. Fake medications mean that unauthorized medications available maybe in the markets, in the black markets, and some of these medications are toxic. Really, they are toxic, and maybe we read in the newspapers that some people affect with these fake medications. I collaborate with Professor Aslam Beg in Pakistan to analyze some of these well-known common medications. It is called these kind of medications for anti-diabetic drugs, and they are fake medication unauthorized from FDA. We found the following. These are the tablets. We did analysis all through this cross section of these tablets. We found using, of course, LAMPA technique and FTIR technique as well. We found this layer, these out layers compositions from titanium and aluminum, and the inside of these layers we found calcium and magnesium and another elements in these compositions. The problem is the titanium and aluminum concentration are more than allowed dose for drugs. They are very toxic, and the dose, the allowed dose is the dose available in these tablets are three times the daily dose of aluminum and titanium, which represent a toxicity according to FDA. And we published this foundation at microchemical journals. At the end, I would like to finalize my talk that LIBS spectroscopy not only allowed us to generate a very high temperature, something like 10,000 Kelvin plasma with a very intense, using very intense laser and high density of electrons, but also gave us an opportunity to make analytical analysis of the content of these elements. Also, LIBS technique can give us an opportunity to analyze medical drugs, environmental samples, and the combination of laser and nano particles enhance the treatment of some type of cancers like breast cancers. Thank you for listening to this brief lecture about our group efforts in this field of laser spectroscopy in many applications, and I welcome to answer if I can the questions. Thank you very much. Dr. Anas.
Thank you, Dr. Walid, for this informative, fruitful presentation. Myself, I enjoyed it very much, and I liked the application in cancer therapy, breast cancer, as it's a huge problem in Egypt, and of course, all other applications are beneficial too. So now we are open for discussion. Let's start with Dr. Rania, as she was trying to ask many times. Dr. Rania, do you have question, please? So, Saifullah Jamal, you can unmute the mic and speak. It's allowed for the participant to unmute.
Thank you very much, madam. Dr. Saab, I Saifullah Jamali, PhD students from University of St. Jamshoro, Pakistan. I really appreciate your work. So I have a little bit questions. That is, how heavy elements from soil may affect our health, and how they enter to our body.
Okay, thank you for your question. Maybe I will say to you, Dr. Safiullah, I am not a doctor, as you know. I am physics, but what I will give you a brief information about what I understand.
Okay.
The existence of heavy elements. Would you please close your microphone? If you have heavy elements in the soil, it will penetrate to the plants, and we consuming plants so that the heavy elements will transform from the plants to our digestion system and will be absorbed, and the size, sometimes the size of these heavy elements are in microns and can be penetrate to our cells in our digestion systems and penetrate to our body, and it will be toxic for us, so for example. This is one of the problem. The other is the heavy elements also will transfer to the drainage water, and this drainage water drain on the main drains. For example, in the Egypt, will go through the Nile River, and will be as a source of pollution for us, and the water will be polluted through these heavy elements. Of course, on the other hand, animals, if animals are available in the field and eat some grass and some plants, the heavy elements will transfer to animals like cows and camels, and at the end it will be in our digestion systems as well. So that in both hands, heavy elements in the soil will be harm our body. This is what I understand.